thank you to my wonderful panelists for joining us today. Ladies and gentlemen, we are very lucky to have over 100 years of experience of analytical development on this panel today, this first panel of the first ADL conclave of started by Express Pharma. So it is indeed very wonderful to host such stalwarts who are all spearheading the analytical development function, taking new and existing molecules from early development to release testing. But recently, the journey has been quite challenging, and for instance, recent issues with nitrosamine contamination have highlighted the various challenges faced by these professionals as they endeavor to deliver key studies faster but with reduced budgets at reduced costs. So let me start with the first question of this panel discussion. Obviously, the topic is current advances in pharma analytical development. What are the trends and technologies? And to start us off, I'd like to go to Dr. P.K. Agarwal. Dr. Agarwal, if you could give us your view on what are the current advances and therefore trends and technologies in analytical development. So we also have Dr. Rao joining us.
Yeah. Thank you, Madam. So it's nice introductions given by Deepti. Appreciate to every members. Yeah, based on my experience when we working in the laboratories, we found that a lot of people nowadays is aware about the nitrosamines related impurities by HRMS estimations and spectroscopy techniques like CM, EDX techniques. QBD approach also a lot of people are aware nowadays. These techniques are day by days now increasing in the laboratories. But apart from these common things which I understand, lot of new technologies which analytical laboratories are applying and should apply in the futures, like 2D techniques, 2D LC techniques, is like good quality of instruments, where the tunables gradient delay systems are there. So these techniques are useful during the method transfer activity purpose, because major challenges in the analytical laboratories after development, the methods, then validations and then method transfer activity will take place in the QC department. QC department means it is far away from sometime the R&D section. So the instruments required good quality of the equipments where that lot of technical things are involved, particularly delay volumes is very key important parameters. I understood that React IR technologies also in R&D centers nowadays using for reaction monitoring purpose. Prep HPLC, a lot of people know, but SFC by using the preparation purification technique is also an advance nowadays in the analytical development laboratories. Freezing dyeing technology also is there, and is very proud to say that Hikal's having a lot of technologies, these type of things, and our teams are using this.
Okay. Yeah, Dr. Agarwal, I'll come back to you. But moving on to the other panelists, Dr. Rao, if you could give us your view on if you had to name 3 top trends and technologies, what would be your choice?
Yeah, thank you, Viveka. Good afternoon, everyone. Sorry, I just joined from other meeting. So, according to me, in the pharmaceutical sector right now, I think there are 3, trends. Right now, I think the most important is the impurities. Like, for example, either it could be genotoxic impurities or it could be a nitrosamine impurities, which is the very hot topic since last 1.5 years. And we have, like for example, we have to procure and do a lot of work on the quantification of these impurities using LCMSMS or GCMSMS. This is one of the high focus area. Apart from the GTIs and the nitrosamines, I think a lot of importance in the pharma to do work on the extractables and leachables. So this is one important area which I feel. And the second area is like, when especially in the generic drug product development, I think we do a lot of reverse engineering, to accelerate the drug product development, to understand how the R&D was manufactured and all. So I think earlier the spectroscopic techniques, like either it could be FTIR or any other spectroscopic techniques, including NMR, we used to use in the development only for the identification purposes. Today, I think like techniques like scanning electron microscopy, which is with energy dispersive X-ray spectroscopy, SEM EDAX what we call, and Raman spectroscopy with imaging, and hot stage microscope. I think these are the multiple tools we are using when we are especially doing the reverse engineering for the drug, during the generic drug development. And the third trend is, as Dr. Agarwal also mentioned, I think the QBD approach, which we are doing a lot, and in the drug development, that is to ensure that the development happens right first time, and we understand the product and the process thoroughly. Of course, we use a lot of softwares for the QBD. One includes the design expert software, Minitab software, etc. So these are the three top trends right now I see in the pharma analytical development area.
Thank you. Thank you, Dr. Rao. And now moving to Dr. Shetty. We've had Dr. Rao, Dr. Agarwal talk about the various trends, but can you tell us about the opportunities that are afforded by these trends, the various benefits of these newer technologies? For instance, new technologies and extractables and leachables and things like that. So, could you give us your view on the benefits of these technologies? Dr. Shetty?
Thank you, Viveka. I would like to add what Dr. Rao and Dr. Agarwal has said, just to complement what they have said, is apart from this, there are increasing demands on the regulatory prospect with respect to the impurities. They're getting more and more deeper, like, first it was genotox impurities, then the nitrosamines, then we had inorganic impurities where elemental impurities had to be quantified, classified, identified in the product. And that's, of course, again, using one more analytical technique, that is the ICP-MS. So because impurities also play a role in contributing to the quality of the product, it can cause an adverse effect if these go unmonitored. So these are the things, and this field has evolved from the time when we started working, where most of the analysis were on non-instrumental technique, all from titration to auto titrator, where everything would be recorded, where the analyst working on it, what he has done, so all that is recorded, and it's there is no room for any ambiguity or doubt due to manual intervention. So this is the way the thing has evolved. And coming to your question, the benefits of the technology which we discussed in the earlier questionnaire is these technologies help us to also investigate if something is gone wrong, if there is an adverse effect. You can go to the trace level, find out what is it that has happened, and you can correct it. Maybe you can go to the root cause and correct. Once you've identified what is the impurity, because of the molecular weight, can narrow down the impurities, find out from where it has come and eliminate that. Again, the quality of the product goes up. So all the manual techniques, like earlier, which I mentioned, had specific optical rotation. All that was very manual. Anyone could see something and record something. Sulfuride ash is again replaced completely by the ICP-MS. So yes, going further, the traceability is good, quality is assured, and the regulatory expectations are also met. And as Dr. Rao also mentioned about the reverse engineering, yes, it helps you to understand how the the innovator has formulated his product. You have to achieve characteristics close to the innovator product. It helps you in understanding what is the amount of excipient or what polymorph he has used with the XRPD technology, the hot stage microscopy. All this helps you to understand what is the form they have used. You can come close to that form and then again reduce the cycle of product development. So these are my views on the opportunities and the various benefits of the new technology.
Thank you. Thank you, Dr. Shetty. And Dr. Vijay Bharathi, in our previous discussions, we've talked about all through the evolution of the analytical development function of pharmaceutical industry. You guys have always been dealing with some or the other contamination coming up, some or the other, impurities coming up. So, and I can see from what the previous three panelists have said that there has been a fair amount of evolution from manual to automated to today you're talking about AI integrated into analytical development. But has there been, are there ways to kind of predict what are going to be the new impurities that you all need to look out for before the FDA catches it, before regulators, before it creates a problem? So have we evolved to that stage, the way we can predict what is going to be the impurity that we need to track today, so that before it gets into the products and medicines of tomorrow?
Yeah, so I think you asked a very good question, I would say. The product development is always a tricky one, and innovator has done it, and people believe generic product development is a copycat. But however, as the days are evolving, and what we see is the gap between innovator development and the generic development is they're coming close, I would say. For example, you can see classical case of this nitrosamine, even the RLD or the innovator was also in the same storm, right? The RLD also had contaminants. They have not recognized that. Now, so I would say it's a continuous learning. There's no, I will not say we'll come to a perfect scenario where we will say that we did everything. So if you see what the regulatory authorities or rather FDA is looking for is they're looking at industry, because we are the one who are developing products. We are the one who are involved in all this method development or the product development. So they want to know from us what could be the probable impurities. And, that's the reason so many regulations have come right from the starting materials to the intermediates, not only days back, people used to, work only on API, but now to the starting material, rather they're going 2-3 steps back to the starting material also for this impurity evaluation. And so since, the problem comes and then the couple of solutions comes, now there is risk assessment, which goes on using, the predicting tools, the QSAR tools, understanding. Now agencies asking from 3 different softwares what kind of impurities may come. And I would say it's a continuous process, and I would say we are better learned today rather than a couple of years back. And I would say integrity in development is a must here. And any kind of shortcuts, like people did use the recovered solvent, people did use the same reactor for the same kind of different products, which has led to this kind of the huge, humongous problem. And I would say even innovator, after developing product, after so many years also, he realizes there is something, which he has not accounted for. So the process-oriented kind of product development, like for example, here we spoke about QBD, which more or less eliminates most of the things. So I think it's a continuous process, and enough tools have come now. So I think we are good to go. But it is again, I would say it is not that the generic product development. It's like the products which have come into generic is also pretty complex. So it becomes kind of not the whole set of battery of experiments may not have gone into the generic development, but I would say it's a continuous process, and we are good now. We have learned from our mistakes. We have learned from whatever crisis has happened. Yeah.
Okay, thank you, Dr. Vijay Bharathi. Moving back to Dr. Rao, the amount of OSS, out of specifications that has come up, that is also a particular challenge that faces analytical development professionals like yourself. So, and therefore it leads to data integrity, non-compliance issues, which have always plagued the industry. So what are your views on that? And do you think automation will help minimize these issues? And if so, then, how are you coping with it? Dr. Rao?
Yeah. So Vivek, I think today, I think, yes, you're right. As you rightly said, I think in the quality area, especially today, dealing with, out of specification results or out of trend results is one of the current challenges, major challenges. And whenever we have the FDA inspections, especially the inspectors are very keen to understand what is the root cause, the assignable root cause, what we call it. Or, if you have a probable root cause, whether it is backed up by proper hypothesis study. So these are the real practical challenges. And also, generally what happens in our industry also, whenever the analytical development develop a good methods, they do validation and all. But one of the, weakness in our system, overall, I'm not criticizing anyone, but I think how we develop the method, we don't put everything into the SOP properly, the test procedure. So there are some gaps. So when these methods, however, if they have transferred in the QC area, but after so since the method is not written very properly, or very explained very detailedly. For example, if I am transferring the method to R&D lab to QC lab within India, I can tell some oral instructions and they will follow. But in Western scenario, if I am transferring the methods to Western countries, they said no, everything you write and give me. I don't just take it like that. If you tell orally, so there are certain gaps. And to answer your point, like how we can minimize the errors, like for example, we have implemented LIMS in our company, across the company from last 2 years. We have implemented international platform, LIMS in our, all our 21 sites in Laboratories. I think this was the one of the major steps we have taken through digitalization, because in the LIMS, what happened, we prepare the masters, and where the methods are, very well written and very well put it in the master documents. Then in the LIMS, what happened, the analysts don't have any choice. Suppose analyst, if he has to weigh 25 milligram, he cannot take by mistake also 30 milligrams or 40 milligrams. It will say the LIMS, you are doing a mistake. So I think I think LIMS implementation has really helped us in minimizing a lot of some of the mistake which can probably happen through a lot of manual interventions, and also like the calculations also. Now, the moment analysts take the weight in the balance, it will automatically go into the LIMS worksheet, and the calculations all will happen through the validated custom fields, what we say. So there are no manual calculations. Again, there is no other checking is required. Everything is done automatically. But last but not least, we have also implemented, to share my experience, a robotic process automation. Like the moment all the calculations happened, we need to have a second checker. So even the second check by the reviewer, by QA, that was minimized, the efforts by putting a RPA, what we call robotic process automation, where if, for example, if in the sequence, if I have not integrated any chromogram, if I have deleted any chromogram, so we have a robotic process automation tool where it will automatically detect and tell you, in this sequence, this was the issue. So all these automations are really helping us. We have invested heavily in the digitalization to minimize the manual interventions and eliminate the manual errors. That will really help in us even in the inspections.
Oh, thank you, Dr. Rao. And I think you've also mentioned, when we discussed this previously, the role of artificial intelligence in now analytical development. Can you expand on that a little bit? Because with everything getting automated.
Yeah. So actually, in our R&D IPDO, so when I spoke with I am in the quality management right now, but when I spoke with my R&D colleagues, so when they developed the methods, they developed a lot of data during the development, and also during the product development. So what I understand from them was the artificial intelligence tool is helping, to pull the data automatically, instead of taking it a lot of manual data, compiling the data, it is helping us to check the trends, if there are any issues, all those things can be automated using the AI tool. I'm not much aware of the details, but that's what I understand. Artificial intelligence tool is helping, and it is also helping in saving a lot of time, because generally we do a lot of maybe multiple experiments. So which experiment is the better experiment? So pull all the data automatically, get some trends and choose the best experiment based on outcome.
Right. Thank you, Dr. Rao. Dr. Agarwal, moving back to you, when we were discussing this previously, you mentioned a very interesting, point of view, where you said that when the regulator picks up on a particular, say, impurity and then mandates and makes it part of guidelines, that you need a particular kind of instrument or instrumentation to kind of detect that impurity. Now, one interesting perspective where you offered that, and you said that, okay, this is all very nice to have it as part of guidelines and to have it as part of protocol, but is it practical to have only that kind of thing mandated? So that brings us back to that question of, new technologies will come to detect these kind of the impurities and things like that. But is it practical to mandate it? And how are companies then dealing with these kind of guidelines to use a particular technology? Are your staff trained on these new technologies, etc., etc.? And what is the kind of capital investment which you need to make to bring in place these new technologies? Dr. Agarwal?
Yeah. Thank you, Viveka. I appreciate Dr. B.M. Rao's communications, which he used the latest technologies in the thought process. Actually, what happened when we are using the latest technologies, definitely a lot of challenges are there. Challenges, particularly if you count simple sex, these instruments are very expensive instruments. Number one. Number two, those people who are run the instruments needs very expertise level of the working. Like suppose Dr. Rao, what he's saying, robotics instruments, robotics way of approach, or when I'm saying 2D LC techniques, if somebody is using, or even HRMS instruments running in the laboratories in R&D center is very tough. So normal people cannot run the instruments. And second point is these machines costs are very high. So if machine costs are very high, then definitely AMC, CMC's costs also very high. So it needs very expertise people's. It's very costly reagents required. It's good type of environments required where the dedicated space is availability is also is important. So if we count challenges in these type of techniques, if you count, number one is expensives. Number two is very expertise scientific staffs required, and third one is because all these things are linked with the IT sections. So definitely normal IT people cannot handle these equipments, and so the persons required who handle the situations will be linked with the analytical persons as well as IT approach. So in my opinion, these type of three challenges are very common in industries, and nowadays people are facing, generally all industries are facing nowadays, because side by side attrition rates also creating a troubles to managers or those who are leaders in the industries, because once we train the person, after training the person will left the organizations, create troubles to all the systems. That's all.
Thank you. Thank you, Dr. Agarwal, for those interesting perspectives. And, while this panel might be only about the current advances in pharma and analytical development, I think this is very important for us to also look at the challenges to implementing these technologies in our labs. So, Dr. Veena Shetty, if I could move to you, how has Reliance Pharmaceuticals and your lab dealt with the kind of challenges that Dr. Agarwal was talking about, where you train a person and there's attrition, etc., etc. So while you might have invested in the capex to put into place these technologies, how are you implementing them to the best of their capacities, Dr. Shetty? And then coming back to you, Dr. Vijaya Bharathi, with the same kind of question.
Yeah.
Dr. Shetty,
Yeah. First of all, I think the base of any analytical method is a good development, a very robust development. Like how Dr. B.M. Rao sounded, like, the method is developed in one environment. It needs to be transferred to different environment. So the first basis, develop the method properly. Coming back to the high-end instruments, like what we discussed, LCMS, like how Dr. Agarwal mentioned, these are all highly sensitive equipments and cannot be given to be handled by anyone. One is operational level, second one is the interpretation level. So both of them need to be in tandem to get the best output of the machine. And yes, what he mentioned is even the maintenance of these machines are high. So it's better to have people who are dedicated to use these machines, trained, and have a full hands-on expertise on these machines. Meanwhile, you can develop a layer because yes, attrition is there. We know about it, so we need to be prepared. So we need to have all-rounders. Like in my team, we see to that everyone knows about each work, though they have to graduate, but from one level to the other level. Like stability, again, method in the method development life cycle. First is the development, then come the stability studies, then you validate the method, then you transfer the technology to the laboratory. So you see to that the method is handled again by different people. So you know the challenges are there. Like it's one person who's developed the method is not biased. So you can eliminate all these problems when the method goes into a different environment, and there has to be no failures. You have to take care of all the aspects. What can possibly cause failure? Maybe not 100%, but then by the end of it, your method is certified if it's workable and reproducible in the other lab. To achieve all this, you need a well-trained staff. Okay, the training is the most important thing. You have to chart out your training. You cannot have all experts at all levels. They need to be built up over a period of time. Okay, so you need to have a proper plan how we are going to take them into a different level. As when they are at the level what is being chalked out to them, they need to get trained and be an expert and move on to another level. So you have a career plan for them. They also look forward to go to the next level. So this is one way of doing it, and this also reduces variability. If everyone is going to be doing everything at the same time, you are going to be guiding them each and every time. So you have sections. You have stability section. You have a development section. You have a validation section. Has to be separate. Has to be unbiased because that's a last milestone for a method, and it's going to be transferred to the different laboratories. If you plan and you have a proper strategy, this can be done because all these problems are not new. We know about it, and what Dr. Agarwal rightly put, people learn and then they want to leave and go to the other organization. Maybe they have learned half, but then it's again for the person who has done this, it's a retraining, again doing all this thing all over again. It's a waste of time for the organization. So might as well have a second level also to take care of any such exigence.
Right. Dr. Rao, I know you need to leave. So before we come to Dr. Vijay Bharathi, can you give us your, your advice to cope with these challenges in a couple of sentences? Because these are challenges which are always going to be there. These problems are not new. If it's one impurity today, it's going to be another impurity. Attrition is always going to be there in the pharma industry. These are very, niche spaces. Dr. Rao, I don't want to take away from your time. So your parting comments.
Yeah. So Viveka, as Dr. Veena also mentioned, what we are also doing is even today, the once upon a time HPLC is a instrument which we used to feel big about in the QC labs. Today, even we have LCM SMS, GCM SMS in the QC labs, to analyze the nitrosamine impurities, and genotoxic impurities, because these impurities are also part of the specification, while releasing the batches. So what we are doing is we are training the people, I know we are prepared for any attrition. We are having good training. We are also keeping a backup, enough backup. And we are also hiring these students, after giving good training. Like we are taking from premium institutes. For example, in our company, we take from campus recruits like NIPER and other premium institutes, like pharmacy graduates and all. And, we train them, and we know very well that, they are very ambitious. They leave after, 2 to 3 years, But still, we always keep a backup. That is the only choice. We can't do anything right now.
Right. So I think that if DRL today trains them and they go to somewhere else, they'll probably come back to DRL in some years anyway. So you might as well train them properly wherever, in whichever company they are, because they will stay as part of the pharmaceutical industry.
Yes, absolutely.
Thank you, Dr. Rao, for spending your time with us.
Thank you, Viveka, for this opportunity. Thank you, everyone.
Thank you. Dr. Vijay Bharathi, all these challenges that are there, and as they said, you're not new. So what is your perspective on how to tackle it?
Yeah, so the new technologies will come. HPLC was also once upon a new technology, and the similar kind of mindset was there. Now, LCMS, once upon a time, like we could count in fingers. Now that has become a commodity. Now, NSA, because of the crisis of NSA, it has pushed every company to purchase these LCMS and GCMS and set up in QZs. So every technology comes and it takes by storm, or it replaces certain technology. So now that we cannot avoid. So the people who use these technology and use these techniques has to be properly trained, and that is the one worry or fear. Usually, the technology gets established over certain years, for example, say 3 to 5 years, then all the problems start coming up, and then we get exposed what could be the pitfalls. So I think FDA looks that we promote technology, rather they also actively participate in certain platforms wherein they push, rather I would say FDA pushes the industry to take encourage technology, new technologies to come in. So you see the history, ICP-MS was pushed by FDA and whatever. So I would say industry never comes up front and say, I have a technology, the product quality would be good. And then based on that, industry always have a very cautious kind of approach. That is regulatory, thanks to regulatory agencies, I would say, in a positive attitude, if I have to say, like what we could not see before, we are able to see now. The new technologies will take its own sweet time to get established, and in that period we will know what are the pitfalls and what are the precautions, what we have to do now. See, I have started my career in mass spectrometry in like 1999. So that was kind of an art kind of thing, now this has become commodity. I see the quality of people who are handling the instruments. They won't understand much about it, but there should be continuous emphasis on, everything is a basic sciences. Whatever applied sciences we are doing, everything is a basic sciences. So emphasis should be given on to the academics also, that people the students who are passing out should have a good sound knowledge on the basic sciences, physics, chemistry, mathematics. So, so when we come to the industry, we can apply these principles, and whatever is the technology, whether it's an XRD or an LCMS or an NMR, lots of advances are happening on NMR also, especially if you see the peptides, every company is doing peptide nowadays. So if you see that NMR is playing a very significant role. Earlier, it was like one NMR in a company used to be there, but now so many number of NMRs are coming up, and FDA is asking certain sameness data, characterization data using these NMRs. So we used to have like one NMR expert in a company. Yes, definitely it's a niche area, but I believe that you have to nurture this. This is very essential for any product development. Rather, it is essential for humans, because we need quality drugs. Yeah.
Right. So, Dr. Agarwal, we were also talking about, in fact, all of this, as Dr. Vijaya Bharathi said, the regulator is kind of pushing the industry to kind of, implement new and use new technologies. So that therefore the impact of regulation on the sector is obviously quite a lot. What is your view on that, and how does industry kind of react to it? Obviously, they have to accept it, but any way to make it a little bit smoother? What has been your experience at Hikal?
Yeah, thank you, Dr. Bharathi and Viveka. I understand what Dr. Bharathi is communicating in the forum. There's a lot of things are now changing. We have also seen same type of situations when the peoples are using simple instruments, and the instruments are only one instruments in industry. Nowadays, same instruments, multiples, and a lot of people's expertise peoples are using. Similarly, regulatory agencies also creating a different type of environment nowadays, and it's definitely impact on the all pharma industries. In my point of view, see, regulatory agencies required always encourage the peoples to use the correct terminologies, correct thought process, correct way of the working. But we have to now use how we have to work. They use they always say that you use certified reference standards, certified thought process, qualified validated automation process, or qualified experts, scientist. So now previously we are using informal way, but now it is formal documentation process is there. So definitely it is more complex is there. Its investment is more. Its time also is there. So all these things are adding the cost of the drugs and the process. A lot of challenges are there nowadays in automation process. Different root causes. OS also is playing the role nowadays, and it's definitely long term. It is benefit to organization, but in my opinion, it's increasing the time factor as well as cost. That is true, and every industries, every lab is facing these problems. Time pressure is more nowadays, and those things previous 15 years, 20 years before, those same things people are doing in relaxed manners now is in complicated manner in documentation system and quality assurance department people's also checking all these documents of ADL section. So same time non-availability of the instrument expertise people's also affecting the things. And as I previously discussed, the instruments are very costly, so AMC CMC costs also very high. So ultimately in final, if I would like to say, time as well as cost is affecting the things.
True. And you bring up cost, and therefore that becomes, the whole debate about sustainability, because you want a certain amount of quality, the regulator mandates it, and we too as an industry want it, because at the end of the day, you're making a medicine which is going into a sick patient's body. So it is a matter of life and death. So how are these new technologies and, sustainable, and what are sustainable from the point of view of, the time that goes into it, the cost that it entails, and therefore what is the, and neither are we left with any options. There's no option to take a shortcut, because as we've seen, the previous shortcuts have left to the impurities of today. So, what is is there any way out of this kind of thing? And how do you all view the technology solution providers in this whole landscape of, a pharmaceutical company trying to get their product out there into the market in the quickest possible time, in the safest possible manner? So how does the technology solution provider play into this mix? Because I've also heard about AMCs and CMCs becoming very expensive. So is there some kind of way to kind of reduce that and partner with technology providers? Dr. Veena Shetty, if you could go to you.
R&D level, maybe you can try to partner with the technology provider, but then that is up to a certain extent. But I think due course of time, it's better that you have everything in house. When the technologies are new, if the commercial labs are having these equipments, initial stage or R&D stage, you can outsource that. But then, these commercial laboratories also are going to take a long time, because they'll have their own set of priorities. And then I think a faster feedback will help you go into the next level of development. So you will only lose time if you are going to outsource. Outsourcing temporarily is good with the solution provider or the technology provider or the commercial testing laboratory. But in the long run, it's better to have all these things in-house to have better control.
Right. Yeah. I remember on our previous discussion about ENL, about extractable and leachables too, you had the same point of view where you would rather do the ENL testing in-house rather than outsource it. So that is
because when like extractable and leachable over here, it's a centralized laboratory where the laboratory caters towards the vaccine department, the gene therapy, the therapeutic protein, the pharma, and the peptide division over here. So you are having all the database over here. The expertise comes only after the experience. So you are having different type of molecules. You are having different type of formulations. But the primary packing material would be same. It would be a vial or a pre-filled syringe or etc. etc. So all this experience will definitely help you to understand and troubleshoot if there is something which is shooting above the threshold level. So and there are challenges with different molecules. Like with the biopharma molecules, we've seen that the leachable, the recovery of the leachable standards, it's a challenge because somewhere they're getting bound to the molecule. So then how to overcome that? So this is a rich experience which is going to help you once if you settle these, or once if you standardize these processes, it's going to be a great path for the future. You can have these set processes for your future analytical work.
True. Thank you, Dr. Shetty. Dr. Vijaya Bharathi, if we could come to you with that, what are your views on sustainability and how do you make it sustainable?
So the scenario, if you see the new technology comes, there are regulatory expectations and the process efficiencies and all. So sustainability, when we speak, for example, AI is one thing which is gripping, there's lots, everything depends on this data science, how much data we have. We have accumulated so much data and so much knowledge, but we are not able to comprehend that. So sometimes, you need an AI to understand, for example, the stability trends you would like to have, or you would like to have something on impurities database and all that. So coming to this sustainability, there are two thought processes. One is that it depends on the pharmaceutical company, how big it is, what kind of, if it is usually what my experience, because I am in right now, not in a product-based company, but I'm in a contract research organization where I've seen that especially the companies which are involved in, if we say that the RLD or the innovator pharma companies, they want to avoid the repetitive jobs. So for that, that is one thing. And so that they want them to focus on the new areas, disease areas, or a new, platforms and all. So now for them, the sustainability is to get new drugs or unmet medication conditions, that kind of medication into the, they want to work on that. For example, you take Novartis, you take Pfizer, or kind of companies. So other set of group of companies which they want to do everything in-house. And when you speak about sustainability, it depends on their way of work, the model, what they want to take it. In my opinion, for example, the data science or AI kind of it, definitely, but if you see the Aragen is front runner for the digitalization, there's lots of emphasis given on that. But we don't develop our own software or like something like that. For example, we depend on, say, Amazon or AWS because they are the masters there. So I am an applicant, I use it. So, so we should draw a line where what we want to do for ourselves or what we want to take help, do a smart job and do that. Now coming to the sustainability part, we should be ahead of what, for example, NSA, like it hit you, right? But you should be in a position like what are the future trends and how you're preparing for yourself. That's very important. It's not that when the need come, necessity come, we start digging it. So rather, it should be a planned kind of future, like what could be the next 5 years, how you want to shape yourself, whether you want to go digitalization or you want to have organically, you want to grow or kind of it. So I think every company should have their own. Coming to our, analytical field, if you see the sustainability is only through, I would say, kind of, because we are in regulated environment, we have to have these systems like LIMS or AIs. Slowly, I think we have to get automation into our this thing. Already it has come. You cannot operate any system or any instrument without 21 CFR Part 11 automatically, whether you like it or not. It has come. So sustainability going forward is in automation and training of the people not to do kind of job where they've not understood fully. Companies give emphasis on the knowledge management and value people, because that's very important, because they are the one who are developing the product. So yeah, these are my thoughts.
Thank you, Dr. Vijaya Bharathi. And I think we've got a very good mix of panelists, because we have, from a pure play pharma company like Dr. DRL, Dr. Rao was there, Dr. Veena Shetty from Reliance, Dr. R.K. Agarwal from P.K. Agarwal, sorry, from Hikal, who's from a very huge manufacturing company, and Aragen Life Sciences, Dr. Vijaya Bharathi, who's from the CRO sector. So we've got a good perspective of different aspects of the analytical development process in the pharmaceutical industry. We have around 15 minutes, so I'd like to, on this panel, so I'd like to go quickly around the panelists and take their key takeaways on what they think are going to be the future technologies and trends which are going to hit this industry. Because as Dr. Vijaya Bharathi said, so can we be prepared for the future? And to all our viewers who have logged in, as well as I'm quite sure that we have quite a few students from premium institutes like NIPER who have joined in. So they would like, if they're just stepping into industry, what would be your guidance to them on how should they keep track of what's happening in terms of new trends and technology, say in this decade ahead? If you could start with Dr. Agarwal, please.
Yeah, thank you, Viveka madam. See, at the very beginning, I have informed about the latest technologies, which Dr. Rao also acknowledged these things. Other panel members also communicate same thing. Actually, see what happened. Previously, we are working on HPLC and GC, or sulfated S, or titrator, or like that. But nowadays, we are working on the GCMS, LCMS, HRMS equipments. Further, if we increase the techniques like 2D LC techniques, multi-column techniques for method development purpose, reaction monitoring purpose for React IR technologies, prep purpose like SFC techniques. So what my suggestion is to all the youngsters and scientists is that we have to upgrade first to keep in mind latest technology. See, Viveka ji, see, we cannot escape from the new development in the system. Like I cannot use now old mobile phone like 15 years before I was using, 10 years before I was using. I cannot say that I will run HPLC in UV lamp only. I have to use PDA. Then I have to use further PDA now converted into again new technologies is there. See, a lot of people know IPDEA techniques are there. See, gradient walls are there previously. Now tunables gradients delay system is there. So I mean to say that name of the technology is not important. One line, one sentence is important. We have to upgrade ourselves, and we have to, with open-minded, adopt it. Open mind, I have to listen my youngsters. They know more than me, and explore in the market and take it. That is more important, and then only we'll be happy and enjoy the life.
Thank you.
Yeah.
Thank you. I think that's a lesson for all of us seniors, where we have to listen to our youngsters.
Yes.
And whether it's in journalism, where I keep learning from my team members, from the young team members on the board, because
I always believe that they know better than me.
Absolutely. So you have to always hire smarter people, right?
Yeah.
If your juniors are smarter than you, then obviously your whole organization benefits.
That's why I'm smiling and I'm enjoying the life.
Very true. Dr. Veena Shetty, your last views before we end this panel. Then
emphasizing on upgradation. We've been having this culture of making one presentation every month by each team member. It can be with respect to the guidances. They read and they understand, that's one thing. And then cross-function also. Cross-functional, what I mean is Most of them are scientists from the analytical chemistry field. So I see to that they understand when we are making specifications, it's for all microbiology, everything. But there are many things, everyone is not a microbiologist. So they need to read, and there are microbiological departments over here. They can go and see how they do it, understand and explain it to the others. We have this every month and it's recorded, like, it's a training also, and it's self-training. So they understand the principle. If it's a new technique which they have not handled, we see to it that most of the time it's something which they have not done it earlier. So the machines are not there over here, they read it, or we many a times we have these vendors who come who introduce new machinery. So I make a point to take them there, understand, learn myself also, because we have started with titration and then now we are moving at this high-end machine. There are many things which we ourselves need to learn before we guide others. So we go there, we learn, maybe not the entire team, but then we split and then we go and we see whenever we have time. We learn there on site, and then whenever the machine comes to us, at least the people are having some idea about what is it, what the machine can do, or what we can analyze, or how we can make use of these technologies. So that is one thing. Then second thing is, I think even if you're going to buy some machine, it's better to closely work with this application scientists from the vendors, because they are very good at operating their machines. You are very good at having different type of products. So this integration is very important. Like the previous session, Dr. Agarwal mentioned, we do have scientists and we do have IT people. But there is a gap in understanding. We do not know anything about the system policies and all. And system policies, again, is one thing which was missed out. It brings about a lot of control in your 21 CFR thing. You do not want a person to delete a data. You do not want a scientist to go and touch the method. You want it to be run in the same manner. You have a lot of controls on this system policy. And even again, with respect to the method development, I think your method needs to be challenged, and it's a requirement. Product life cycle management, method life cycle management, because there are so many, there are going to be so many variables after you have launched the first 3 batches or exhibit batches. Over a period of years, your batch to batch variation in your excipient, in the people who are handling the machines or the people who are going to analyze. So this life cycle management also needs to be a part of the system to have a good or a consistent quality. So
Thank you.
My
Thank you, Dr. Shetty. Thank you, Dr. Shetty. Dr. Vijaya Bharathi, your views, please. Your takeaway messages.
Yeah. So how we can, what would be the future and what kind of, so according to me, it all depends what kind of products will come in the future. For example, the small molecules development or the discovery is slowly slowing down, and it is other complex products are coming into the picture, whether it is the chemistry or the biology or integrated development. Would you say the large molecules, since the disease are progressing in such a way that the normal antibiotics or this thing has gone into past, and now new products are coming. So now when the product is complex, the characterization or the analytical skills has to be complex. Or I would say the era would be for hyphenated techniques, wherein one instrumentation or one platform may not help you. You got to have a hyphenated technique there, wherein you have to have maybe it's already there, like you have, for example, CE machine linked to MS or NMR, you do. So I think era would be for hyphenated techniques plus the automation, and the next would be, since there's so much knowledge around, there's so much data around, the AI is definitely going to take the center stage, and for the orphan drugs or very unmet conditions kind of drugs, wherever the innovator drug industry is taking us, for example, oligonucleotides or the peptides or different type of therapies, whatever comes in, you have to fit in, because analytical is a support function for all this. So I believe like we have a good we have a good scenario where we have a different kind of instrumentation and technologies, and now maybe we have to bother from material science, maybe we have to bother it from different allied streams, from the clinical or something like that. So I believe mostly it would be hyphenated with automation and of course all these new IT whatever is coming up like AI or kind of this thing. All these has to be in place. It will, for example, mass spec is going to go still long. Now single quadruple is getting replaced by the QTOF or the Orbit Wraps, HRMS. Next other platforms also are going to come into picture. So yeah.
Thank you. Thank you, Dr. Vijay Bharathi, Dr. P.K. Agarwal, Dr. Veena Shetty, and Dr. P.M. Rao, who had to leave us. Thanks so much for all those, deep analysis over the past 1 hour on the current trends and techniques and technologies. And as we said, depending on the product, analytical development is going to change and will be changing in tune with the products. So I'm very glad that we had this panel of experts to guide us on that and put ADL conclave in the right note and put us all in the right mood for the next sessions on.